In this lesson, we are kicking off with May-June 2017, paper 21. So looking at multiple choice, mixed topics, let's see what we get. Question 1, starting at the beginning of your syllabus, which characteristic do all living organisms show? And see how they've even put that all in bold. So they really want to drive home the fact that this is the characteristic that all of them show. So breathing. Not all living organisms breathe. Excretion? Now that sounds like an eligible answer. Let's check the other ones. Photosynthesis and tropism. Well, those both seem that they are rather plant-based. So definitely not all organisms. So yes, we can confirm that the B, excretion, that is a characteristic of all living organisms. No matter the size, no matter the style. All living organisms go through excretion. Question 2. The diagram shows some animal cells as seen under the microscope. Animal cells. What will be present at X? So we can see a whole lot of cells all surrounded by their membrane. These are animal cells, remember? And then we can see the nucleus in each of them. And then we've got little dots of things that we can't see enough detail to see what they are. So what will be present at X? One cell membrane, one cell wall, two cell membranes, or two cell walls. Well, remember that cell walls are only in plants. So we can cross out B and D straight away, because those ones are both plants only and we're dealing with animal cells. So now we have to think about what's going on here. And we have to remember that each and every cell is surrounded by its cell membrane. They don't ever share a cell membrane. So if we zoomed in on X, we would see those two cells next to each other, each with their own cell membrane. So we would have two cell membranes at X. If we had a line over here, let's say this is Y. At Y, there is only one cell membrane, because we're on the edge of only one cell. But because X is between two cells, we have two cell membranes. Question 3. Each of the diagrams shows an area of cytoplasm. So we're not looking at an entire cell here, just a piece of cytoplasm, which is from a prokaryote. Remember, your prokaryotes are things like your bacteria, so simple organisms. And the big thing there is that they don't have any membrane-bound organelles. So if we look at these, we can see rough ER, spelling atrocious, so rough endoplasmic reticulum is showing here in A. In B is one with a teeth inside. These are the mitochondria. And in D, we have chloroplasts. Now, all of those are organelles. All of them have membranes around them, so they can't belong in a prokaryote. If we look at C, this is our circular DNA. That DNA that isn't packaged into chromosomes in a nucleus, and that is a feature of a prokaryote, which confirms C is our answer for question 3. For question 4, we're moving on to tissues, away from just a single cell. We have a diagram that shows a section through a root, and we are labeled P and Q and R. So now we have to figure out what are the levels of organization of the labeled structures, P, Q, and R. So if we think about it nicely, we're looking at a root. What are the fluffy things on the outside of a root? Those are root hair cells. So P here is our root hair cells. Okay, so P would be coming in at the cell. Then it's between the organ and the tissue. And remember that we go from cells to tissues to organs. So Q, looking at the cortex of that root, this is representing the tissue, where we have lots of cells that are doing the same thing. So Q is our tissue, and our organ is something that is performing the function, the root, absorption of water and mineral ions. So R would be our organ. So then we have to look, where does it say PRQ? It says it at B. That matches our answer. Question 5. Which characteristics are correct for both, in bold, osmosis and diffusion? Now remember that you can scribble on your papers, because of course you're just sending away that answer sheet. So really get to know these papers, get involved in your questions. Okay, so for both osmosis and diffusion, require a partially permeable membrane. Think about the definition. Diffusion, well, deodorant diffuses across a room. If you spray it in one corner, that doesn't need a membrane. Osmosis, that has to be across a membrane, but is not required for both. Require a concentration gradient, from a high concentration to a low concentration, from a high water potential to a low water potential. And water potential is representing that concentration of the water, so yes, we require a concentration gradient. And then, are energy consuming processes? No. Both osmosis and diffusion are passive. Neither of them use energy. So definitely a cross on that one. So where do we have cross tick cross? We have it here at C. Characteristics correct for both osmosis and diffusion is just that concentration gradient. Question 6. Which diagram shows the structure of DNA? Well, when we look at those, we can straight away cross out C and D, because we know the bases are in the middle. We're not stuck on the outside. C and D just look far too odd. Okay, so bases in the middle. How do we say which one between A and B is the DNA? It's all about who bonds to who. Remember with our bases, okay, we have very specific pairings. We can either have AT or we can have GC. A and G, A and C, it's never going to happen. They're actually the wrong sizes and the wrong shapes. They don't fit together at all. So if we look at A, we see here AC, AC, TG. Those don't happen. That's wrong. If we look on B, we have AT, AT, TA, CG, GC, AT, GC. Those fall into the pairs that we're allowed. So our answer for the structure of DNA is B. Our bases are in between our backbones. A only bonds to T, G only bonds to C. Question 7 moves us on to enzymes with one of our classic graphs. The graph shows how enzyme activity is affected by temperature. So we have our rate of reaction, and then we have our temperature. So if we do experiments at 0, we get a little tiny bit rate of reaction. 20, we would have more things happening. If our experiment was at 40, we'd have lots happening. If our experiment was at 60, nothing would be happening. So, how can the change in activity between 40 and 55 be explained? So, this piece. Why is this happening? It's all about denaturation. Because it's too hot. Now that enzyme has denatured, it's changed shape. So let's have a look at our possible answers. Heat has killed the enzyme. No, I'm afraid the enzyme wasn't living to start with, so that is not true. The enzyme has been used up. Nope, because each of these different temperatures is a different experiment with new different enzymes. So we can't use them up. The reactants are moving faster. Well, yes, but that would cause further. That would use more interactions and more reactions. So that is not going to explain a decrease. That would cause this increase. So we certainly hope that D is going to be right. The substrate is less likely to fit into the active site. Yes, they change shape. So now they're not going to be complementary anymore. So they're not going to fit together. We completely agree with them. The answer is D. So what comes next? 4 test tubes are set up as shown. Which test tube contains the most carbon dioxide after 1 hour? So let's see what we've got going on. We've got water and aquatic plants in the first 3. Then we've got snails in A and D. Okay, A and B, we have no light. C and D, we have light. So, in C, light, we have photosynthesis. So we're going to have lots of oxygen given off. We're going to have very little carbon dioxide. So C is out. D, we're going to have respiration. So we could have carbon dioxide here, because he's going to be using oxygen, giving off carbon dioxide. So let's have a consideration about what's happening in the dark. Now, in the dark, the plants are also going to be respiring, but not photosynthesizing. So here we're going to potentially have CO2, and here we're going to have CO2. But let's look at this. Here we've only got plants. Here we've only got snails. Here we have plants and snails producing CO2. So who's going to have the most? Well, the more sources of CO2 is going to give us the more CO2. So our answer is A. Question 9. The graph shows daily carbon dioxide uptake and transpiration by the plant Agave americana. The plant is adapted to live in very dry conditions. So, with the black dots, we have carbon dioxide. So carbon dioxide uptake is high, and then it drops, and then it goes up again. And then transpiration is all over the place a little bit. Okay, so transpiration is higher at night than during the day. There is something funky going on here, because normally transpiration would be higher during the day for our plants. Okay, so what can be concluded from this graph? More stomata are closed during dark periods. Well, no, because we've just said the other way around. Okay, stomata have to be open for transpiration, and we've got more transpiration at night. More transpiration, see, it's higher in the nighttime hours. So they are not closed in the dark periods. More stomata are closed during light periods. Well, now that makes sense, because that would decrease that transpiration in the light. We'll check the other 2 to play safe. There is no carbon dioxide uptake during the dark periods. Well, that's not true at all. There's lots of carbon dioxide there. There's no water uptake during light periods. Water uptake? That happens in the roots. We can't see that at all from this graph. All we can see is water loss. So, definitely confirmed, more stomata are closed during the light periods. So this plant is doing very funky things with his carbon dioxide and transpiration in order to adapt to these very dry conditions. So then, question 10, sticking with our plants. In plants, which ions are used to make amino acids? So remember, your amino acids build your proteins. And proteins are made from carbon, hydrogen, oxygen, and the special one, nitrogen. So, which of these ions is going to give this special nitrogen? The nitrate. So it's nitrates that the plant is going to use to build amino acids, to build proteins. Question 11. The following symptoms can be caused by a dietary deficiency: bone pain, dental problems, fragile bones, and skeletal deformities. A lack of which nutrient is most likely to cause these symptoms? Iron, protein, vitamin C, or vitamin D. Remember, the calcium and your vitamin D are the ones who are very closely linked. Calcium contributes to bone structure. It is vitamin D. A lack in vitamin D can cause bone pain, dental problems, fragile bones, and skeletal deformities. Question 12. Statements 1 to 4 describe stages in the development of cholera. So we have chloride ions are secreted in the gut. Osmosis causes water to move into the gut. The infected person becomes dehydrated. Toxins are produced by the pathogenic bacteria. What is the correct sequence of the 4 stages? So what is it that happens first? So the first thing that's got to happen is that those toxins have to be produced. We're not going to have any symptoms or any response unless those toxins are produced. So there's our 1. What do the toxins cause? Those toxins cause chloride ions to be secreted. And that changes then the water potential, and then water moves by osmosis. And it's that loss of water that causes the dehydration. So we should be going 4, 1, 2, 3. So our answer is A is out, B is out, C matches the order that we've got. Sticking with dietary things, talking about teeth. Question 13 tells us that the diagram shows a human tooth. Which statement best describes the function of this tooth? It bites into food, it cuts into food, it grinds food, it tears food. And this is a classical shape for a molar, which is all about grinding food. So you do need to be familiar with those teeth as you're learning them. Mechanical digestion and teeth are an important aspect. Question 14. The graph shows the effect of pH on the activity of 3 different enzymes. So we have enzyme activity against pH, going from very acidic. There's our 7 of our neutral, going up to basic. So enzyme X, enzyme Y, and enzyme Z. And you can see each of them have a different optimum pH, and that is going to relate to what they do and where they are. So the table shows the pH of different parts of the alimentary canal: the mouth, the stomach, and the small intestine. So which enzymes in the graph are likely to be protease enzymes? So let's line them up. Mouth with our pH of 7, so that then is Y. So Y is the mouth. Stomach, pH of 2. So there's our stomach. And then small intestine, pH of 8.5. Yes, it does line up with Z. So our protease enzymes chop our proteins into smaller pieces, then they chop them into amino acids. Remember, we've got 2 zones for that to happen. Where does that happen? It happens in the stomach and the small intestine. It doesn't happen in the mouth. So Y is out. X and Z, or Z only, X and Z, 2 zones, plus we know that our protease, our chopping up proteins, starts in the stomach at this low pH. So our answer is B. Moving on to question 15. Skipping all the way back to plants, the diagrams show cross sections through a root and a stem. X marks the spot in the root. Then our stem is all around outside. So which labels are correct? Where is our phloem? Where is our xylem? So our W is wonderful because X marks the spot and X starts for xylem. So W is xylem. Which begs V our phloem. So we can knock out C and D straight out. So it's about these ones. Which way round does it go? Now think about it. We come through the root and into the stem, right? So if our xylem is in the middle on the root, it's got to be in the middle on the stem. Can't just suddenly jump to the outside, which means that Z must be our xylem on the middle there, and Y on the outside is our phloem. So Z is xylem, Y is phloem. So phloem, xylem, our answer is C. Question 16. Which processes are used by root hairs to take up ions and water? Now, how does water move? Water always moves by osmosis. So we can take out B, we can take out C. Ion uptake, active transport or osmosis? Well, osmosis can only be water, so it's definitely not ion uptake. And if you think about it nicely, we're pumping those ions into the plant, definitely active transport for ion uptake. Question 17. Jumping back to plants and talking specifically about transpiration again. So, which of the following increases transpiration? Air around the leaf with high humidity. High humidity means lots of water. It means less of a gradient. So that one is going to decrease transpiration. Air molecules around the leaf with less kinetic energy, meaning the air molecules around the leaf aren't going to move so much. So again, we're decreasing that gradient. Decrease it. An absence of light falling on the leaf. Well, no light would mean no photosynthesis, would mean stomata closed, would mean no transpiration, which certainly is then going to decrease transpiration. Water molecules in the leaf with more kinetic energy. Well, the more they move around, the more likely they are to diffuse out of the leaf. So yes, that one is going to increase transpiration. Question 18. The diagram shows a human blood cell. What is its function? Antibody production, fibrinogen production, oxygen transport, or phagocytosis? So, this blobby thing here is a very funky nucleus. So, this must be a white blood cell. It is a particular one. This is your phagocyte. This lobed nucleus is what we call it. And that is your classic thing that tells you that is exactly what that cell is. So if we look at these, oxygen transport and fibrinogen production, we can take those straight out. Those are not even white blood cell functions. So antibodies or phagocytosis, what does a phagocyte do? It does phagocytosis. So 18 is most definitely D. Question 19. What is a disease-causing organism called? An antibody, a host, pathogen or a phagocyte? Well, we certainly know it's not a phagocyte because we just talked about phagocytes a moment ago. Antibodies are things that we produce in order to fight infection. A host, well, that would be us, and it would be where the pathogen is infecting. So the disease-causing organism is the pathogen. I'm hitting question 20 and exactly halfway through this paper. What is a function of the cartilage in the trachea? Gas exchange, prevents collapse of the trachea, produces mucus, or traps bacteria. So remember, the cartilage in the trachea, this is a structural thing. It's giving that strength to the trachea. So gas exchange, no, that's all about the alveoli. Preventing collapse, sounding good. Producing mucus, that is our goblet cells. Trapping bacteria, that's the mucus that was produced. So yes, definitely the cartilage in the trachea is helping to prevent collapse. So we shall continue with the rest of this in the next lesson.